And we'll go back to our screen with biochemistry. And yesterday we talked about carbohydrates. So we're going to continue with discussing some of those groups that we mentioned that are part of biochemistry. The next group that we're going to discuss is very different than carbohydrates. The next group we're going to discuss is lipids. And one of the reasons that they're very different than carbohydrates is carbohydrates have a very specific chemical identity. We said carbohydrates are polyhydroxyl, meaning they have a lot of OH groups. Ketones are aldehydes, meaning they have a carbonyl group, a carbon with a double bonded O. If we look at lipids, they don't all have the same type of chemical structure, but they do have something in common. Lipids have mostly nonpolar bonds. Well, in organic compounds, nonpolar bonds are bonds between carbon atoms and another carbon atom, so carbon to carbon bonds, whether they're single bonds, double bonds, or triple bonds. But also remember, carbon and hydrogen make a nonpolar bond. So lipids are molecules that have mostly nonpolar bonds. So this tells us they're going to be mostly carbon atoms and hydrogen atoms, not a lot else. And since they have mostly nonpolar bonds, they're mostly hydrophobic. They don't mix very well with water. Think of oil and vinegar dressing. Oil is a lipid, vinegar is mostly water, and they separate into layers. So lipids have really large areas that are nonpolar bonds, and then they have a very small area with polar bonds. So overall, most of the lipid molecule is hydrophobic, but there's a little part. Which part? Well, that part that has the polar bonds, that's hydrophilic. A lot of times you can tell a lipid if you touch a lipid or if you have a lipid in your hand, because it has a kind of a greasy, oily feel. So that's typically a giveaway that it's a lipid. There are different categories of lipids because, as I mentioned, they don't all have the same chemical makeup. So one category or class of lipids is something that we call steroids. And you've probably heard of steroids before. If we look at the chemical structure, steroids are made of 4 interlocking rings of carbon. So let's see what we mean. So here's cholesterol, which is a steroid. So remember, a ring of carbon is a carbon where is a group of carbons where the last carbon in the chain is bonded back to the first one. So when we look, here's a ring right here with 6 carbons. Here's another ring with 6 carbons. Here's another ring with 6 carbons. And here's a ring with 5 carbons. Now, these are interlocking rings. So how do they interlock? Well, because some of the carbons are part of 2 rings. If we look at these 2 carbon atoms right here, these 2 carbon atoms make up the right side of this ring, and these 2 carbon atoms make up the left side of this ring. So that's how these 2 rings are connected or locked together. These 2 carbon atoms make up the upper right of this ring, and these same 2 carbon atoms make up the lower left of this ring. And then finally, we have 2 carbon atoms here. They make up the right side of this ring, and those 2 carbon atoms also make up the left side of this ring. So steroids all have these 4 interlocking rings of carbon. Now, like in most of the diagrams that we've seen so far, the lines represent chemical bonds. But remember that carbon needs 4 bonds. So we don't see 4 lines around most of these carbons. There are 4 lines around a few of them. Here's a carbon right here that has a line, a bond down to this carbon, 1 to this one, 1 to this one, 1 to this one. But this carbon only has 1 bond attached to it. Where are the other bonds? The other 3 bonds are bonds to carbon atoms. So the bonds to to hydrogen atoms, the bonds to hydrogen atoms are not shown in this diagram. Now, anything that isn't a hydrogen attached to a carbon is shown. So here we have an OH. The hydrogen is shown just because it's bonded to that oxygen. So everything else in this molecule in cholesterol is a carbon or a hydrogen atom. So carbon to carbon bonds are nonpolar. Carbon to hydrogen bonds are nonpolar. So this whole big section over here on the right and towards the top, this is all nonpolar. This is all hydrophobic. The only part of this molecule that's polar and hydrophilic is this little section down here where we have this OH group. So this is kind of the general characteristic that we see in lipids. Almost completely nonpolar and hydrophobic. A small section, much smaller, that's polar and hydrophilic. So if we look at some of the steroids, in the upper left, we have what's sometimes called the steroid nucleus, which is those 4 rings. Now notice in this version, they're not even showing the carbon atoms. So we saw some examples like this the other day. So remember, the end point of each line is a carbon atom. And again, all the carbon atoms have enough hydrogens that they have 4 bonds. So we have these 4 interlocking rings, and then there are 2 CH3s. You might remember that the CH3 is one of those functional groups we talked about when we discussed organic chemistry. It's the methyl group. So this is considered to be the nucleus. These are the kind of the basics that we find in almost every steroid. Occasionally, one of these methyl groups is removed. So here's a diagram of cholesterol, what we saw just a moment ago up above. Here's progesterone, a hormone in the female reproductive system. Notice instead of an OH like cholesterol, we have a double bonded O down here. And we have another double bonded O on a carbon up here. And our chain stops at this point right here. It doesn't extend further. And then if we look at testosterone, hormone in the male reproductive system, we've got that double bonded O again. And notice that ONE is a hint that this is a ketone. And remember that ketones are molecules that have that carbonyl group, that double bonded carbon. So in testosterone, we have this, and then we have an OH up here. So notice it's very similar to progesterone. Estradiol, cortisone, aldosterone, all of these chemicals are examples of steroids. And we might think of steroids as being found in people, but notice that animals called vertebrates, fish, reptiles, amphibians, mammals, birds, all make steroids. There are some other animals like insects that make some steroids, and even plants make steroids. So we don't want to get the impression that steroids are only found in people. So steroids are one type of lipid that we see. Another type of lipid that we see is something called fatty acids. And fatty acids are just really organic acids that have that carboxyl group that we talked about before, that COOH functional group. And remember, with that carboxyl group, we said that the key is we have a double bonded oxygen and an OH, a hydroxyl group, on the same carbon. So here's an example of a fatty acid. And since they have the carboxyl group and really everything else is carbon and hydrogen, fatty acids are sometimes called carboxylic acids. So here's our COOH over here on the left. So we have a carbon with a double bonded O and an OH on the same carbon. So all fatty acids have that. The fourth bond, notice that there are 3 bonds to oxygen atoms. The fourth bond is to the next carbon in the chain. And then we have a chain that's just carbon and hydrogen after our COOH. Now, if we look at the bonds between all the carbon atoms, and if we look at those carbon-to-carbon bonds and they're all single bonds, this is called a saturated fatty acid. When something is saturated, it's full. After it rains a lot, sometimes the soil has as much water as it can hold, and we say that it's saturated, and if it continues to rain, the water just runs off the soil and forms puddles. This has as much hydrogen as it can hold. If we look at all these carbons after our COOH, they all have something in common. They have two bonds to other carbons. They're bonded to the carbon in front of them in the chain. They're bonded to the carbon behind them. And then their other two bonds are connecting the carbon to hydrogen till we get to our last carbon, and it's a methyl group, CH3. So when we look at this molecule, it's saturated. It's got as much hydrogen as it can hold. In an unsaturated fatty acid, un at the beginning of a word means not. In an unsaturated fatty acid, there's one or sometimes more than one double bond between the carbons. So if we put a double bond between the carbons here, a couple of things to notice. First of all, notice the two carbons on either side of the double bond. If we look on either side of the double bond, these carbons only have one hydrogen. Because remember, carbon atoms always need 4 bonds. So if we look at the carbon on the left of the double bond, it has a carbon ahead of it. It has 2 bonds to the carbon behind it. So 3 bonds so far, 1 to the carbon in front of it, 2 to the carbon behind it. So it only needs 1 bond to a hydrogen. So notice these 2 carbon atoms do not have as many hydrogens as they could hold potentially. If they didn't have a double bond, there would be room for another hydrogen atom. So that's why this is called unsaturated. So in terms of the formula, if these 2 fatty acids have the same number of carbons, the unsaturated fatty acid will have 2 less hydrogen atoms. Everything else would be the same. They both have 2 oxygens, they both have the same number of carbons, but every time we add a double bond, we lose 2 hydrogen atoms. So if we had 2 double bonds, the formula for this unsaturated fatty acid would be 4 less hydrogens than the one up on top. Now, the other thing to notice is where we have a double bond, there's a bend in our molecule. And this is crucial if we have saturated fatty acids, because the molecule is relatively strong, we can pack them together. But since the unsaturated fatty acids are bent, we can't really pack them very close together. They've got these bends that make them kind of spread out. So unsaturated fatty acids are much more loosely organized. And this is going to be crucial in a few minutes when we look at some of their properties. Because usually when we look at fatty acids, whether they're saturated or unsaturated, we don't find fatty acids, by themselves in cells. Usually fatty acids are combined with other molecules to make bigger things. So fatty acids can be used as building blocks for larger lipids. So let's look at some of the larger lipids that we can build by using fatty acids and combining them with other molecules. So first of all, let's look at how we can change a fatty acid that's unsaturated to a fatty acid that's saturated. So here we have something called a polyunsaturated fat. So remember, we talked about polysaccharides the other day. Poly means many. And so in this case, there are 2 double bonds. I don't know if I would call 2 many, but when we talk about polyunsaturated fatty acids, they usually have 2 or 3 double bonds. So notice the carbons here that are part of the double bonds only have one hydrogen, like we talked about a moment ago. What we can do though is we can break those double bonds and we can add hydrogen. So now in our product, notice this reaction is going downward on the page. In our product, all these red hydrogens are hydrogens that weren't here before. And this is how they take peanut butter, the way that it comes directly from peanuts, and this is how they make it into processed peanut butter. If you've ever opened a jar of natural peanut butter, there's some solid material down on the bottom, and up on the top, there's some liquid. Well, that liquid is unsaturated fatty acids. And so what they do when they process peanut butter, they take those unsaturated fatty acids and they break some of the double bonds and they make them saturated. And this is called a hydrogenated fat. So if you look on a jar of, say, Skip or Jiffy or something like that, unless it's the natural version, because some of those brands do have natural versions, but if it's just regular Skippy or Peter Pan or Jif, one of the ingredients is hydrogenated vegetable oil. So that means they added some vegetable oil, and we'll see in a moment, oil is a type of fat. They added some vegetable oil, and then they broke the double bonds, and they added hydrogens. Because when we look at saturated fatty acids are usually solid. Polyunsaturated fatty acids or even monounsaturated fatty acids are usually a liquid. So if we have an unsaturated fatty acid, it's usually a liquid. So for instance, vegetable oil is mostly unsaturated fatty acids, corn oil, sunflower oil, safflower oil. So now that we can think about that, let's look at how we can use these fatty acids. So here's a fatty acid, doesn't matter whether it's saturated or unsaturated. This particular one happens to be unsaturated that they're showing, or happens to be saturated, sorry, that they're showing us, but we could use an unsaturated one. We're going to combine a fatty acid with an alcohol. And it's called a long chain alcohol because it's got a lot of carbon atoms in it. So remember the other day we talked about ethanol, which had 2 carbon atoms, and methanol that had 1 carbon atom, and rubbing alcohol or isopropyl alcohol that has 3 carbon atoms. Notice this one has 30 carbon atoms. It has 29 CH2 groups in a row, and then it has a CH3. So there's so many carbons, that's why this is called a long chain alcohol. What we're going to do is the same thing we did with carbohydrates. The way that cells make bigger molecules is by a dehydration synthesis. So we're going to take the OH off of the carboxyl group. So we're going to remove the OH from the fatty acid here. And this is pretty common. This happens in a lot of reactions. The fatty acid will lose its OH. We're going to lose the hydrogen from the hydroxyl group on the alcohol. Now notice there's lots of other hydrogens that are found in CH2s and CH3s. We're always going to take the hydrogen from here. This hydrogen will react with the OH group. These other hydrogens won't. So if we take this OH group off, notice we have a problem. Our carbon atom here only has 3 bonds. It has a bond back to the next carbon in the chain. It has a double bond to this oxygen, that's 3. But if we remove the OH, this bond is gone. We know that carbon needs 4 bonds. If we take this hydrogen off, our oxygen atom right here only has 1 bond. It has a bond back to this carbon. Oxygen atom needs 2. So what a cell does in this reaction is it removes the OH from the carboxyl group. It removes the hydrogen from the hydroxyl group. So OH and H2O, that makes water. That's why this is a dehydration synthesis. Then where the carbon is missing another bond and the oxygen is missing another bond, they're bonded together. So if we look at our product, notice our product, we have CH3, CH2, 14. exactly the same as up here, so nothing happened over on this part of the molecule. We have our carbon with our double bonded O. Our OH is gone. So now we have a bond to an oxygen, this oxygen over here. And this oxygen is connected to CH2, and there are 29 of those CH2s, and then CH3. So nothing happens over in this part of the reactants, nothing happens here. All the action is in this area. So this molecule is a wax. So remember our basic quality that we said that lipids have. They're mostly nonpolar and hydrophobic. So areas that are nonpolar and hydrophobic just have carbon and hydrogen. So here's a big section, 14 CH2s and then a CH3. This section is all nonpolar bonds. It's all hydrophobic. Over here, there's a section that has 29 CH2 groups and then CH3. So this section is all nonpolar. It's all hydrophobic. So the only part that's polar and hydrophilic, the only part that will mix with water, is this little section right in here where we have the carbon and the two oxygen. Those are polar bonds between carbon and oxygen. So that makes this part of the molecule polar. It makes it hydrophilic. Now notice the other parts are much bigger. There's 15 total carbons here. There's 30 total carbons here. Yeah, there's one here. So the polar part is much smaller, but remember, that's what we said we expect when we look at lipids. Now in real life, this molecule usually kind of folds into a U shape. The turn of the U is right where this polar carbon and oxygens are located. And then the two parts of the U that stick up are where we have all the carbons. Notice that the two parts of the U aren't necessarily the same size. This one is 15 carbons over on the left. This one is 30 carbons over on the right. So we have a kind of a U-shaped molecule. So we've really got one carbon, maybe two out of a total of about 50 that are polar here. So this molecule is very strongly nonpolar. So because wax is so nonpolar, it's used a lot of times for waterproofing. So wax is found in the ear canals of a lot of animals, because if water just kind of drips in from the outside, this can cause an infection. The wax also has an important function in the ear canals. You know, you probably felt the wax in your ear canal at some point when you've been cleaning it out, and the wax is kind of sticky. So besides keeping water out, you know, particles that are in the air, small insects that fly into your ear, things like that, might get stuck in the wax. Now, because it's waterproof, a lot of plant leaves have a waxy coating on them. Sometimes if you rub your fingernails along the leaf, you can kind of scrape off that wax. So why would a leaf have a waxy coating? Because it's waterproof. And for plants, they actually have one of the same issues that people and other animals do, especially in hot, dry weather like we're having now. They can get dehydrated. They can lose water. So one way to avoid dehydrating is if the leaves are covered with a waxy coating. That wax prevents the water inside the leaf from evaporating. And beeswax in honeycomb, you can kind of tell by the name, has wax in it. So a lot of times wax is used for waterproofing. But notice I named a few places where waxes are found. But if you think about something that's in the ear canals of certain animals, it's coats leaves, it's in beeswax. That's not necessarily a lot of locations. So let's look at some types of lipids that are much more common. Let's look at a type of lipid called a triglyceride. And tri gives you a hint, think of tricycle, that there's going to be three of something. Triglycerides are sometimes also called neutral fats. And there's two basic categories of triglycerides. There's what we call fats, and there's what we call oils. Now, we make fats and oils the same way. We have a molecule called glycerol. So let's look at glycerol over here. And notice glycerol has an OL ending. So that's a hint that glycerol is an alcohol. And glycerol has 3 carbon atoms. It's written kind of up and down here in the peach color. And each carbon atom has an OH on it. And then the other bonds are hydrogens. And what's going to happen to make a triglyceride is we're going to react each of these OH groups, each of the hydroxyl groups on alcohol, with a carboxyl group, a C double bonded O, OH, on a fatty acid. So we have 3 fatty acid chains. Each one of them is going to react with one of the OH groups. And like we saw before, remember before when we made our wax, we had an alcohol and the alcohol lost a hydrogen atom. And we had a fatty acid and the fatty acid lost an OH. So here we're going to lose an H and an OH each time these combine. So ultimately we're going to make 3 molecules of water. And where we lose our hydrogen, this oxygen is now missing a bond. Where we lose our OH, this carbon is now missing a bond. So similar to what we just did with our wax, we're going to make a bond between this oxygen and this carbon. So our product, kind of visualize the carbon skeleton for a moment and ignore all the hydrogens and oxygens. Our molecule that is our product kind of looks like the letter E. Now it has really long strands sticking out to the side. Because notice our glycerol here is only 3 carbons down. And now we have a bond between this oxygen that lost its hydrogen and between this COOH that lost its OH. So we have a bond. So we have something that looks like the letter E. And notice in a cell, this happens step by step. So the first thing that the cell does is it adds 1 fatty acid, and then we have a monoglyceride. And then it adds a second fatty acid, and then we have a diglyceride. And then we have a 3rd fatty acid, and we have our triglyceride. Now, if we look at fats, fats contain mostly saturated fatty acids. Now, remember a few moments ago when we were talking about fats and saturated fatty acids, we said with saturated fatty acids that they have as many hydrogens as the carbons can hold. So the molecule, the chain here is kind of straight. So if we have a fat, we can pack fats very closely together. And because we can pack them very closely together, fats are generally solid. And fats usually come from animals. But remember when we were talking about fatty acids, if we go back for a moment, we said if we have an unsaturated fatty acid, that puts a bend in the molecule. And because of that bend, we can't pack them together very tightly. So if we have unsaturated fatty acids in here, we have a bend and we can't pack them very closely. So if we have a lot of unsaturated fatty acids, we don't have a fat, we have oils. And because we can't pack the fatty acids in an oil very closely together, they're more spread out, they end up making a liquid. And oils usually come from plants. Now, even though fats are more solid, oils are more liquid, it's still, both molecules have the same type of function. They're both used to store energy. And they're generally used to store energy for long periods of time. We talked about starch and glycogen the other day, and usually starch and glycogen are used to store energy for briefer periods of time. Now, notice if we look at fats and oils, our usual rules aren't true. There are a few types of oils that actually have so many unsaturated fatty acids, that have so many saturated fatty acids, I should say, they're relatively solid, like coconut oil and palm kernel oil. Usually oils, usually things that have unsaturated fatty acids, are healthier for you. And the reason is because when they get inside your body, saturated fatty acids can be converted into cholesterol. And so saturated fatty acids can raise your cholesterol. You could have a diet where you don't eat very much cholesterol, but if you eat a lot of foods that have saturated fatty acids, your diet could still be causing your cholesterol level to go up. So oils are much better. So olive oil is one of the ones that there's the most evidence for being healthy, but corn oil, sunflower oil, safflower oil, things that are liquids. Usually the thicker the liquid gets, the more saturated fatty acids it has, so the less healthy it is. So we find lots of these types of materials. So if you think about what happens when you eat a steak, if you see like, a little bit of white around the edges of the steak, or sometimes we say that, a hamburger is marbled, there's little pieces of white in it. That white that you're seeing isn't cholesterol. Some people think it's cholesterol, but that's actually fat. It's actually triglycerides that mostly have saturated fatty acids. So you can sometimes see fat. Now notice you're not seeing individual molecules. You're able to see it because there's so much of it there, it's visible. Even though this looks like a lot of atoms, the individual molecules are still tiny enough you couldn't see them with your bare eyes, even with a very good microscope. Now, notice that this drawing is not completely accurate in the sense that glycerol has 3 carbons, and in the middle of our fatty acid here, it shows ... meaning, you've got some more CH2s. The fatty acids that are found in fats, like in the human body or in some kind of food you're eating, usually have 10 or 12 or 14 or 16 or 18 carbons in this part. So this part of, I said you could think of this like a letter E. This part is 18 carbons long frequently. This part is only 3 carbons high. So this molecule has really long, whatever those parts of the letter E are called there. So we want to keep that in mind. Now, sometimes we can combine fatty acids with glycerol, and we can make different types of compounds besides triglycerides. So let's look at something called a phospholipid. And in a phospholipid, we're going to take glycerol and we're going to combine it just with 2 fatty acids. So at 2 of the spots up here, we're going to combine our glycerol with the fatty acid, but then we're going to combine it with a phosphate group at the 3rd spot. So what we see is something like this. So here's our 2 fatty acids. Notice that they're both saturated. And keep in mind what we said, even though we call this a straight chain, it's not exactly straight. It's got that little kind of zigzag pattern. Here's the area where we have the O that was part of the OH in glycerol connecting to the C double bonded O that was part of the carboxyl group in the fatty acid. So here's our 2 fatty acids. And then on the 3rd carbon, it's combined with a phosphate group, which often has an R group attached to it. A lot of times that R group actually has an amine in it somewhere, but not always. So this whole kind of bluish part of the molecule is hydrophilic. It's got a lot of polar bonds, carbons bonded with oxygen. And really, you could probably include this carbon double bond too. This green part is more hydrophobic, carbons and hydrogens, carbons and hydrogens, carbons and hydrogens. When we look at phospholipids, phospholipids are crucial. Remember, one of the properties of living organisms that we talked about back in chapter 1 is that living organisms are made of cells. Well, when we look at cells, every cell is covered by an outer structure called a plasma membrane, or it's sometimes called a cell membrane. If we look at this plasma membrane is made of 2 layers of phospholipids. Mostly. There are some proteins in there too, and a few carbohydrates, but mostly 2 layers of phospholipids. So this means for every cell, and this is not just in the human body, but for every cell in every living organism, there are 2 layers of phospholipids surrounding the inside of the cell. So there are lots and lots of phospholipids out there. They're very important. Later in the next section, after we get done discussing chemistry, we're going to talk about cells. And one of the things that we'll spend a lot of time discussing is the plasma membrane of the cell. Because since it's on the outside, the plasma membrane is what lets things into the cell or out of the cell, or keeps things where they are on the outside or inside and doesn't let them pass. So the chemistry of the plasma membrane is very important. So a lot of times when we look at a phospholipid, the phospholipid will be represented like this. This round area represents the polar section, and the polar section is usually called the head. So this reddish round or orangish round area is the polar head. These 2 long green strands are represented by the 2 kind of rectangles. And these are the nonpolar tails. Notice if we see a bend, what does that tell us? It tells us that this polar acid, tells us that this fatty acid has a double bond here. Remember that double bonds put a bend in the molecule. If we had another bend partway down here, then this would be a polyunsaturated fatty acid. So when we look at lipids, they don't all have a similar chemical structure. So they're not like carbohydrates in that sense. What do all lipids have in common? Mostly nonpolar molecules, mostly hydrophobic. And why are they hydrophobic? Why is the molecule nonpolar? Because most of the bonds are nonpolar, because there's big chunks of the molecule that are just carbon and hydrogen atoms. Now, another category of molecules that we find in living things is proteins. And proteins are an example of what are called macromolecules. Things that are microscopic, micro means small. Things that are macroscopic are big. So macromolecules are big molecules. Now, we've already talked about one type of macromolecule. We just didn't call it that. Polysaccharides, like cellulose and glycogen and amylose, those are macromolecules. Those are large molecules. And large molecules all have something in common. They're built from small units. Now, proteins are built from smaller molecules called amino acids. And amino acids have 3 different functional groups. If we look at the name tells us 2 of them. They're amino acids. So they have the amino group. Remember, that was a nitrogen with 2 hydrogens attached. They're acids. So they have that carboxyl group. Remember, we just talked about fatty acids a moment ago. So they have that C with a double bonded O and an OH. So this amino group and this carboxyl group are both attached to a central carbon. The central carbon has a hydrogen atom, and then it has an R group. And the R group is different in every amino acid. Now, if we look at proteins, there are millions and millions of different proteins. There are 20 different amino acids. And as I said the other day, one way to think about this is proteins are words, amino acids are letters. So how do you spell a different word? Well, you either use different letters or you take the same letters and you put them in a different order. So how do you make a different protein? Well, you either use different amino acids or you take the same amino acids and you put them in a different order. Rat and tar are two different words. If you have two proteins that have the same amino acids but in different orders, the proteins will have a different shape and they'll work differently in the body. A lot of times when we look at the amino acids, they're divided into groups based on their R groups, because some of these R groups are polar, some of these R groups are nonpolar, some of them are ionic. Some of them have another NH2 group in them. Some of the R groups have another COOH. The simplest R group is just another hydrogen. So when we look at amino acids, you could think of amino acids as spelling a protein. The amino acids are the letters, the protein is the word. So how is a cell going to combine amino acids? Guess what? A dehydration synthesis reaction. Same type of reaction that we talked about with carbohydrates. Same type of reaction that we talked about with waxes. Same type of reaction that we discussed with triglycerides, with phospholipids. When cells make bigger molecules, they pretty much always do it by a dehydration synthesis. Now, if we combine two amino acids, we make something called a dipeptide. Remember with carbohydrates the other day, we talked about disaccharides. So how are we going to do this? Well, the carboxyl group is going to lose an OH. Now notice that's the same thing that we saw when we looked at making a wax a minute ago, or making a phospholipid a minute ago, or making a triglyceride. And the amino group is going to lose one of its hydrogens. Notice that we've got another hydrogen here. We could have a hydrogen in this R group. When we combine two amino acids, the hydrogen is always going to come from that NH2. It's not going to come from any place different. So here's an example of a dehydration synthesis. And obviously, if you think about how much time we spent discussing dehydration synthesis reactions, you know that you're going to see some questions about these on the next test, on the chemistry test. So here we have two molecules of an amino acid called glycine. And glycine is the amino acid that I mentioned that has an R group that's just another hydrogen. So one glycine loses an OH from its carboxyl group. The other glycine loses a hydrogen from an amino group. So that makes a molecule of water. So remember what we said when we talked about carbohydrates. If a cell needs this water, the cell can retain it, but otherwise the water flows into your blood and either gets eliminated through your urine or through your sweat. Or if other cells in the body need it, they might remove water from the blood. So when we take this OH off here, we have a carbon that only has 3 bonds. Minus this oxygen, we have a bond to this carbon, and we have a double bond here. So we have a carbon that only needs 3 bonds, only has 3 bonds. We know that carbon needs 4. Over here, we have a nitrogen. After we remove this hydrogen, we have a nitrogen that's got a bond to a carbon, a bond to a hydrogen, and nitrogen needs 3 bonds. So we make a bond between these two atoms, between the carbon atom that lost its OH, between the nitrogen atom that lost its H. And this molecule is a dipeptide. This is called a peptide bond. Now, if you go back and you think about this peptide bond in terms of what we talked about a few days ago with bonds, this peptide bond is a type of polar covalent bond. We have a covalent bond between carbon and nitrogen. They share electrons, that's why it's a covalent bond, but they don't share electrons exactly equally. The nitrogen hogs them a little bit. So that's why it's a polar covalent bond. Now, we can do the same thing. Down on this end, we could take an OH off and we could add another amino acid over here. Once we get past a dipeptide, we don't usually call the things that we're making tripeptides or anything like that. If we just have a few amino acids in our chain, we call it a peptide. Peptide is about the size as the oligosaccharides that we talked about. If you remember, an oligosaccharide has between about 3 and 8 monosaccharides in it. So a peptide is about the same length. If we get a much longer chain of amino acids, it's called a polypeptide. So kind of like a polysaccharide, if you will. So why do we keep using PEP? Well, because the first protein that was actually studied in very much depth is a protein called pepsin that comes from the stomach. And one of the reasons it was studied is because it's very easy to get pepsin out of the stomach. So it was easy to study in that sense. You didn't have to do anything too complicated. You can even take the pepsin out without killing an organism if you do it very carefully. So now, what we want to keep in mind in particular about proteins is something that we mentioned with molecules in general. Even though they're small, even though they're so small we can't see them with a microscope, molecules have a three-dimensional shape. They have a structure. And for proteins, the way they're going to work depends on their structure, their shape. Change their shape and they don't work, or they work differently. So there are 4 levels of structure in a protein that ultimately determine its three-dimensional shape, its three-dimensional structure. The first one is the primary structure. And the primary structure is what we've mentioned once or twice already. The primary structure is just the order of the amino acids. So if the first amino acid is glycine, and the second one is leucine, and the third one is threonine, and the fourth one is phenylalanine, that's our primary structure. That's the order of the amino acids. Now, the secondary structure is caused by some chemical bonds called hydrogen bonds. And so far we've talked about covalent bonds that are polar. We've talked about covalent bonds that are nonpolar. And we've talked about ionic bonds. So we haven't really talked about hydrogen bonds yet. And we'll get to those maybe later today, but if not later today, tomorrow, when we talk about water, because hydrogen bonds are easiest to visualize in water. Well, we can have hydrogen bonds between one amino acid and another amino acid. And these hydrogen bonds basically twist our protein, and they twist it into 2 main shapes. One of these shapes is called an alpha helix. So an alpha helix is the basic shape of a spring, or if you ever played with a slinky when you were a little kid. It's that basic kind of twisting spiral shape. Or they can form a pleated sheet. A pleated sheet is like a folded sheet of paper. So on this diagram, these little circles represent the amino acids, and the different colors represent different amino acids. So in the secondary structure, here's an alpha helix, here's a spring. We've kind of stretched out our spring. Here's our beta pleated sheet. Now the beta sheets and the alpha helix got their name because the first 2 letters of the Greek alphabet are alpha and beta. That's how the alphabet got its name. But when biologists first started studying protein shapes, they figured they were going to discover lots and lots of different shapes. So that's why they gave them these. And then the third letter of the Greek alphabet is gamma. So they were going to name something gamma. The problem is, these are the main two shapes. And then most of the other shapes are kind of unique. We don't see the same exact shape over and over again. So biologists quit naming things by using the letters of the Greek alphabet. Now, think about these two basic shapes. So suppose you have some amino acids that start out pretty far apart. We might have the first amino acid and the 10th amino acid. Once we start twisting our protein, once we start folding it up, now we have amino acids up here and down here that used to be pretty far apart, but now they're close together. So now these amino acids that started out far apart, but they're closer together, now these amino acids can interact. And sometimes this happens because of what we call hydrophobic interactions. Sometimes it happens because of what we call disulfide bridges. And there are lots of other types of chemical interactions also. But in most cases, what happens is this folds our protein into a globular shape. So here's a protein folded into a globular shape. Now, if you look within this globule, you can see some areas like right in here. If you look at this zigzag line, this is a pleated sheet. But if we look down here, we see that spiraling helix. So we can get this globular shape through a mixture of alpha helixes and pleated sheets in different parts of the protein. And some proteins don't fold up into this glob. Most of them do, but some of them kind of stay stretched out. And if they stay stretched out, we call them fibrous proteins. And there are some fibrous proteins that you've heard of, like keratin. Does anyone know where keratin is found in the human body? It's found in at least 3 distinct locations. Does anybody know what one of those locations is? Well, keratin is found in your hair. It's also found in your nails. And if you think about your hair and nails both grow out of your skin. Keratin is actually found in your skin. So think about your skin versus your hair versus your nails. Those 3 parts of your body have very different texture. They don't feel exactly the same. So that's a good example of how the same chemical can lead to different results, depending on some things that we'll talk about later. Now, finally, if we look at some proteins have a quaternary structure. Remember, we said a long chain of amino acids is a polypeptide. Some proteins are actually made of several polypeptides. And a quaternary structure is how multiple polypeptides twist around each other. Now, because it's how these polypeptide chains twist around each other, it's only found in proteins that have more than one polypeptide chain. So remember a few days ago, we talked about a protein called hemoglobin, and we talked about how it played a role in a disease called sickle cell anemia. Well, hemoglobin is actually made of 4 polypeptide chains. So hemoglobin has a quaternary structure. It's how these 4 chains twist around each other. Think about it like, the wires at the back of your computer or the back of your entertainment center, where you might have a wire from a DVD player and a wire from a TV and a wire from, different things, and they always seem to get all twisted. So this twisting is intentional in proteins. So if we have 2 polypeptide chains, like we see down on this bottom example, we can have a quaternary structure. So if you look carefully, notice that this kind of light purplish is the same shape as this. So this is one polypeptide chain. And then this darker bluish is a second chain. And they're kind of wrapped next to each other in a very specific pattern. So as long as proteins have the correct shape, they can work just fine. Notice, as long as we get the amino acids in the right order, it might seem like the shape is not that big a deal. But if the shape is wrong, even though the amino acids are correct, it can lead to pretty serious consequences. There's a disease that you might have heard of called mad cow disease. And mad cow disease is a disease that affects the brain. But even though it's called mad cow disease, it can affect the brain of a person. And mad cow disease is caused by a protein. And the protein has the normal order of amino acids. But even though it has a normal order of amino acids, they fold up into the wrong shape. And because they fold up into the wrong shape, this means that this causes this disease, which is often fatal if you get it. It's very rare for people to get mad cow disease, but you could get it if you, if you eat a cow that's infected. So proteins have to have the right shape to work. So why wouldn't a protein have the right shape? Well, one of the things that can happen is we can denature proteins. De means away. So if you denature a protein, you take away its nature. It doesn't work the way that it's supposed to. Now, to denature a protein, you have to break the peptide bonds. And remember, the peptide bonds is what connects one amino acid to the next amino acid. And when you do this, this destroys the primary structure. A good example of denaturing a protein that you've all seen is albumin. Albumin is a clear, thick liquid protein, and it's found in eggs. So think about that clear liquid that's kind of gooey that you see when you crack an egg open. However, if you cook it, it denatures the albumin, and the albumin is now that solid white material. And once you denature a protein, you can't get the original protein back again. You've broken the chemical bonds, you've broken the connections between those amino acids. So they're all separated now. So if you take an egg that you fried and you put it in the refrigerator and cool it down, you're not going to get an egg with liquid white. It's going to stay solid. That change is permanent. This is actually one of the main dangers that can happen if you get a high fever when you're sick. A high fever can denature proteins. And if you denature proteins that are important for the body, and those proteins quit doing their job, you could potentially die from a high fever. You can also denature proteins by changing the chemical environment, by making it more acidic or more basic, or by adding certain other types of chemicals. A good example of how we can do this on a temporary basis is getting what we call a perm, to either straighten your curly hair or curl your straight hair. If we look at this tertiary structure in hair, that tertiary structure is caused by chemical bonds between two keratin molecules. So we have some gray lines here that represent keratin molecules. And there are all sorts of bonds between these. Now, the bonds that get broken when we give your hair a perm is we break some of these bonds that are disulfide bridges. Disulfide bridges are some of these yellowish bonds. So we break those. And when we break those, the two keratin molecules come loose from each other. And when they come loose from each other, they kind of slide back and forth. And when they slide back and forth, this means that your hair that was straight, we can put it in curlers and it'll be curly. Or your hair that was curly, we give it another chemical called a relaxant and we can straighten it out. Now, there is a problem with this, and that is when the new keratin grows out, when your new hair grows out, it grows out in your old configuration. So you have curly hair, we straighten it, it doesn't stay straight forever. There's another problem. When you put chemicals on your hair, it can break these new connections between the keratin molecules, and your hair gradually goes back to its old configuration. Your straight hair that you had curled straightens out. Now, what type of chemicals do we put on our hair? Well, most people use a type of chemical on their hair called shampoo. And shampoo can actually break the bonds between these two strands of keratin so that they go back to their original shape. You can usually buy like special shampoo for perms, but keep in mind, special shampoo for perms is usually just watered down shampoo. It doesn't have as much of the cleaning ingredient in it. So that's why it doesn't cause your permed hair to go back to its original shape. You know, baby shampoo that says that it doesn't cause tears actually is very similar. Baby shampoo is usually watered down so that it doesn't have as much of the soap, the cleaning agent in it. So that's why it doesn't typically cause you to cry if you get it in your eyes. So keratin is one type of protein we find in the body. It's a fibrous protein that doesn't take that globular shape. Like we mentioned, it stays as a long strand. Besides keratin, another fibrous protein that's very important is something called collagen. There's so much collagen in your body because keep in mind, you're really bigger than most animals. We don't think of ourselves as being large, but more than 99% of all animals are smaller than people. But if you could somehow dissolve every chemical in your body except collagen, you'd have a perfect outline of the shape of your body and every organ in your body, every bone, every blood vessel, every muscle, the brain, because there's collagen everywhere. Notice that collagen is used in a lot of wrinkle treatments. Since collagen is found in your skin, if you have wrinkles, you could think of a wrinkle as being like an indentation in the skin. What we do with some treatments for wrinkles is we inject collagen under your skin. So it's kind of like patching a hole in drywall. We basically fill it in with collagen. We make it kind of puff out. So that's one possibility. So we have a lot of fibrous proteins in our body. Now, on the outer surface of our cell, we have some proteins that act as receptors. So over here, this green molecule is a receptor. We'll talk about these others. We're going to come back to this diagram later. But a receptor is kind of like a loading dock, except it's not exactly like a loading dock at a company. If you were at a business and they have a loading dock, a truck pulls up to that loading dock and they unload whatever. If we look at cells, you could imagine that every color here is a different type of protein receptor, and protein receptors are specialized. So if you could imagine a business where, imagine a grocery store and there's a loading dock where a truck pulls up that is the truck that has the milk. And there's another loading dock where a truck pulls up that's the truck carrying the bread. And there's another loading dock where the truck pulls up that has the fresh fruit. Cells have lots of different receptors for lots of different things that they need from the environment. And the receptors, remember what we said just a moment ago, how do the receptors know what to pick up? Well, it's not a conscious decision. A receptor can only pick up something that has the right shape to attach. So if we have a receptor like this green molecule that has a little kind of triangle shaped divot, only a triangle shaped thing will fit into it. So now these receptors aren't perfect. Sometimes what we see is a virus or a bacteria has a shape that maybe kind of looks a little bit like this triangle. So sometimes viruses, every once in a while bacteria, but more commonly viruses, can attach to a receptor and get into a cell. So here's the SARS-CoV-2 virus. Notice that's actually the name of the virus. It's SARS-CoV-2. COVID-19 is the name of the illness that it causes. But SARS is severe acute respiratory syndrome. And CoV-2 it was the second virus of this type discovered. You might remember when you see a picture of this virus, it has these big spikes that stick out. And these spikes are actually proteins. So the spike has a stem, and then it has an area that's sometimes called the head. And there's a little part of the head, this little part right down here. And this little part of the head has the right shape that it attaches to something called an ACE2 receptor protein. ACE2 is a chemical that's normally found in your blood, and it helps to control your blood pressure. Well, this spike protein has a little part that's the right shape to attach to this receptor protein. And when it attaches, there's another molecule. So notice this is our virus up here. This is a cell in your lungs down here, the gray. There's another molecule that has something called a protease. And the protease enzyme cuts off this top kind of darker orange section of the spike. And ultimately, this allows the virus to get into your cell. So because it attaches here, your cell thinks it's something called angiotensin converting enzyme. That's what ACE stands for. So why? Because it's got a shape that's just a little bit similar. So we'll talk in more detail when we talk about cells, but notice this is why typically for cells, viruses only infect certain types of cells. They can't infect every cell in your body. Because if we look at, say, a cell in your lung, like we see here, and we compare it to a cell in your heart, or a cell in your kidney, or a cell in your brain, or a cell in your muscle, cells in different organs have different types of receptors. So when a virus can attach to a receptor, it can only infect cells that have that type of receptor. It can't infect other types of cells. So if we look at the virus, for instance, that causes the flu, that virus has a shape and it can attach to receptors in your, what we call your mucous membranes of the respiratory system. So kind of your nose, your throat, your lungs. So what's one of the symptoms of the flu? You might get a sore throat, you might cough, you might get congested. But the virus that causes the flu can also attach to receptors in your muscle cells. So one of the symptoms of having the flu is your muscles might feel sore and achy. So different viruses cause different problems because of the cells that they can attach to and get into. So we'll look at viruses in more detail when we think about cells.
Is it bad I'm having a hard time understanding this stuff? Probably just watch the videos again, read the
Okay.
notes. But
And keep in mind, for some of these things, we're going to come back to them in more detail later. So when we come back to them in more detail later, sometimes this part will make a little more sense because we'll have to go over this again.
okay.
So what's another thing that proteins can do? Some proteins are hormones. I know you've all heard of something called insulin. You might not be aware, but insulin is actually a hormone. It's a receptor. Now, it turns out almost all the cells in your body have insulin receptors. And So insulin is made in one of the glands of your body called the pancreas. And the pancreas puts insulin into your blood, and insulin circulates through your blood. And when it's flowing through your body in the blood, it can attach to a receptor. And when insulin attaches to a receptor, this is crucial because this allows that cell that just picked up the insulin. Now that cell can take glucose out of your blood. Brain cells don't need insulin. They can always take glucose out of the blood. Muscle cells that move your body don't need insulin. They can always take glucose out of the blood. But most of the other cells in your body need insulin first. So if you don't make enough insulin, one of the problems that can occur is ultimately right after you eat a meal, your blood levels of glucose may be high, but then a couple hours later, they may be very low. Now, why would they be low? Because if you don't make insulin, your cells can't take glucose out of the blood. So ultimately, when you go to the bathroom and urinate, a lot of the glucose comes out in your urine. So there's not enough glucose in your blood. So you can have low blood sugar levels. Now, some proteins are carriers, are transport proteins. They pick up something and they carry it around your body. For instance, if we look at hemoglobin, that molecule we talked about that's involved in sickle cell anemia, hemoglobin actually has 4 chains of amino acids. 2 of them are identical. They're shown in darker purple. 2 of them are identical. They're shown in lighter purple. But hemoglobin has something called a heme group in it. And heme means iron. So in your lungs, hemoglobin picks up oxygen. And the oxygen attaches to the hemoglobin part of this molecule. It attaches to the heme part, to the iron part. So the oxygen gets loaded in your lungs. So we have 4 hemoglobin groups in one molecule. We can pick up 4 oxygens. Then when your blood circulates around your body, when it goes to your brain or your heart or your muscles or your lungs, it drops off the oxygen so that other cells can use this oxygen. And then once it unloads the oxygen, so if this is blood that's going to your heart, it drops off its oxygen. And then later it gets sent back to the lungs to pick up more oxygen. So your blood continually makes a circle. It basically goes to the lungs and picks up oxygen. Then it goes someplace else and drops off oxygen. And it does this over and over and over again. Now, one of the main types of proteins that we're going to talk about this semester is enzymes. And if we look at a typical cell, whether we're thinking about a human cell or some other type of cell, typical cell has about 10,000 proteins in it. And almost all those proteins are enzymes. And what those proteins do is they regulate chemical reactions or metabolic reactions. And even though they regulate them, enzymes generally regulate them by speeding them up. A lot of the metabolic reactions that happen in your cells would happen without enzymes, but there would be a problem. Without enzymes, they would be so incredibly slow, they wouldn't happen fast enough to be useful. So enzymes can speed up these metabolic reactions. And enzymes a lot of times have very long, complicated names. Like one is called phosphofructokinase. However, once you take a chemistry class like biochemistry and you learn the rules, the good thing is that a name like phosphofructokinase tells you exactly what an enzyme does. So that's the advantage of that. We won't look at enough chemistry this semester to learn the rules. We'll talk about a few enzymes and we'll describe what they do. But we want to keep in mind anytime we look at a chemical reaction, there's an enzyme that kind of controls that reaction. A lot of times the enzyme will be written underneath the arrows for the reaction. But even if you don't see an enzyme written there, there is almost always an enzyme. So we want to keep that in mind. So remember the other day we talked about if you don't have an enzyme called lactase, you can't digest lactose, so you'll be lactose intolerant. And since animals make an enzyme called cellulase, you can't digest cellulose, so it doesn't provide you with nutrition. It provides the fiber in your diet. So when we look at some enzymes, here are two examples. So we have a molecule called pyruvate. And notice that if we look at pyruvate, pyruvate has a carbon with a double bonded O and then an O with a negative charge. When we have an acid, and an acid loses its hydrogen, and the hydrogen leaves behind its electron, we see a structure like this. We see a carboxyl group with a C double bonded O and an O with a minus charge. So pyruvate is something that started out as pyruvic acid, and then it lost its hydrogen up here, so now it's pyruvate. Well, an enzyme called pyruvate decarboxylase. Remember, the carboxyl group is the COOH. So D always means away. And the Mg and the TPP up here are two things that this enzyme needs to work. So we'll talk more about that a little bit later. But so the pyruvate decarboxylase takes off what's left of the carboxyl group. So if we look, notice we still have our CH3, we still have our carbon with our double bonded O, and we have to replace this. So we're going to replace it with a hydrogen. And we have something called acetaldehyde. Now we're going to take off the double bonded O, and we're going to add a hydrogen to it, and we're going to add another hydrogen. And the enzyme that does this is something called alcohol dehydrogenase. And again, it needs something called NADH to work. So these are two examples of enzymes. So enzymes always have long names. So for today, we just want to keep in mind, what does an enzyme do? It speeds up a metabolic reaction and it kind of controls it. Some people have compared an enzyme to a traffic cop out in an intersection when the stoplights are broken. If you have somebody standing there directing traffic, it usually goes more quickly than if you just have everybody stopping and kind of starting on their own and deciding when to go. I always think of enzymes as being like my social studies teacher when I was in junior high. If you went to any of the dances at my school when you were in junior high, the social studies teacher would make sure that you got out on the dance floor, because she would grab a guy and a girl, kind of put them together and say, oh, you two need to dance with each other, and throw you out on the floor. That's kind of what an enzyme does. An enzyme gets the reactants together and forces them to make a product. So the enzyme does a lot of the work so that the reactants don't have to do it themselves. So are there any questions about proteins or lipids after today? And as I say, keep in mind, we will come back to a lot of these topics and we'll look at them from a slightly different perspective. So hopefully when we look at things from a couple of different angles, it will make more sense. The other thing that can sometimes help is, I know it's difficult to understand if we haven't talked about it, but if you read the textbook, or if you're using another book, if you read the other book, the material on kind of similar topics, ahead of time. Right now we're doing chemistry. If you look at it ahead of time, you won't understand everything, but then sometimes when you get to lecture, some of the things that we're talking about will make a little more sense because you won't be seeing them for the first time. Well, if there are no questions, I'll stop screen sharing. And are there any questions before we leave today? Okay, if not, I will let you go and I will see you tomorrow morning. And tomorrow we'll finish up chemistry, so plan on having the chemistry test Monday most likely.